welcome back all. We'll continue from where we left in this cell signaling course. If you recollect, we were discussing how receptor histidine kinase system works absolutely fine in microbial world. Rather, I should say that they are heavily dependent on receptor histidine kinase signaling for meeting their need while they are communicating with the environment or communicating between each other. There we saw that how two-component system requires essentially two components, one which was we saw as a receptor and one which was the response regulator. In today's class, we will see more of the details of this two-component system which is prevalent in the microbial world. And also we will see what are the derivatives of the two-component system which exist in the microbial world to meet all signaling needs. So we will discuss first the basics of the two-component system, where a simple presence of a sensor kinase is required and something as a response regulator will be required. That makes it a canonical two-component system. So the first component which is here is the histidine kinase, receptor histidine kinase or the sensor kinase. If you recollect, we said and discussed that the signal which is coming from environment that is being sensed by this receptor, the presence of the signal is being sensed by the receptor. That is why it is also referred as sensor kinase. And a rough domain arrangement was such that there is an extracellular ligand binding domain, then you have a internal dimerization and histidine phosphorylation domain, and then you have a catalytic activity which is associated with the receptor, and this catalytic activity is nothing but the kinase activity which uses this histidine residue in this DHP domain and phosphorylates it utilizing an ATP molecule. So when this receptor binds to a signal, it starts to communicate inside through response regulator, the other component of the signaling. First component is the sensor kinase or the receptor, second component is the response regulator or some sense effector which we have discussed earlier. So the basic need of a response regulator is to have a domain which can generate an output. The simplest response regulator would be something which has this aspartate residue on which this phosphate group can be transferred. So here the histidine residue acquired this phosphate group by use of ATP and its hydrolysis. However, in case of this aspartate residue receiving this phosphate group is a phosphotransfer event. So this is also called phosphotransfer. And by virtue of the function entailed in this minimal domain, you would see an output. Often such simple form of the response regulator domain mediates protein-protein interaction and thus may affect the localization of one protein and affect the interaction of this protein with the other interactor molecule. Now the other type of response regulator could have an additional domain, and this domain can bring lot of other activities. It could be an enzymatic activity, it could be a transcription factor like activity. So what will have, what this response regulator will have, in addition to the minimal receiver domain where this aspartate residue is ready to receive the phosphate group, there is an additional domain. This, if I refer as additional domain, the property of this additional domain is going to define how the response will be moving forward. And as I said, this could have a catalytic enzymatic activity, or it could have a transcription factor role, or many more. So basically in the canonical two-component system, if you talk about only the receptor here, one of the two components, the receptor or the sensor kinase. So as I said, it has a domain which should identify the signal, then the dimerization domain, and then the catalytic activity domain. What I noticed and what I told you in the previous slide is that Generally in the two-component system, the phosphate group moves from this histidine residue to the aspartate residue. This is how it is present, and we saw in the previous slide that the histidine is present in the sensor kinase and aspartate is present in the response regulator. However, there could be variations in the receptor itself, and it may possess both the histidine residue and the aspartate residue. So in such a scenario, what happens that phosphotransfer is happening within the molecule. So first, upon sensing the signal by this domain and the catalytic activity by this kinase domain, histidine is phosphorylated, ATP is utilized. Once this phosphorylation event has happened, the phosphotransfer happens to the aspartate residue which is present within the kinase. So what can drive this movement? The mere presence of this aspartate residue or the overall arrangement? Also, in such a scenario, the movement of phosphate from the internal aspartate residue on the sensor kinase will be a challenge, because we generally notice the phosphotransfer from histidine to aspartate. So in simplest words, you can say that in such a scenario, the movement in the forward direction will be happening from histidine to aspartate residue. If there is no suitable next molecule available, it will be reversed back. So it will be kind of terminating the signal. So if you look at this domain arrangement of the sensor kinase, what you realize that there are conserved functions of these two domains. What all different functions which are mediated by this ligand binding? There are different types of ligand binding domains which have been characterized. Some of them have this motif which is called JAF motif, and these motifs recognize cyclic and linear form of the GMP molecule. This will be one of the molecules which you will see more in detail, that how this affects so many functionalities of the microbe. Then there could be additional domain which is called PAS motif containing domain. This domain is important for recognizing small gaseous molecule, the redox potential changes, and energy or light dependent changes. So basically some of these abiotic molecule will be sensed by the PAS domain which is present in the ligand binding region. Another domain which has been found often in these receptors is the HAMP domain, which helps in the chemosensation when there is a presence of another bacteria. Remember that we saw that how microbe, or in that case we saw dictyostelium was moving towards a signal of cyclic AMP. So here also there could be a signal which will be coming from another bacteria, and the other bacteria will be responding to that. Now if we think in simple terms, so you can have a canonical, if I say two-component system, so you have a sensor kinase which will have the histidine residue which is phosphorylated, and this phosphate is transferred to the aspartate residue which is in the response regulator, and if I say response regulator as RR. There could be a sensor kinase which not only has the histidine which is phosphorylated, it has an internal aspartate residue which receives the phosphate group from histidine. In this scenario, how a response regulator will receive phosphate group? That is a challenge, and we will see how that is solved, and what happens to scenario where you have a sensor kinase of this sort. This often is not part of the canonical two-component system, and that is where you have hybrid two-component system. So let's see what exactly will be different in hybrid two-component system. So as we discussed earlier, you have a histidine or sensor kinase. This will bind to a ligand through its ligand binding domain. The histidine phosphorylation event will take place because of the catalytic activity, the kinase activity which is here, and then the phosphotransfer will happen internally onto this aspartate residue. Always remember that the movement of phosphate follows histidine to aspartate, then aspartate to histidine, and so on. Even in the reverse direction, when the phosphate is moving, it becomes aspartate to histidine, then histidine to aspartate, and so on. So, because internally aspartate residue is phosphorylated, it needs to move this phosphate group on another histidine, and that is where you find next set of molecule which are called histidine phosphotransferases or HPTs. So, the hybrid two-component system will involve another component which is histidine phosphotransferase. So, you can say that it is no more a two-component system, rather a three-component system, right? But this solves the deadlock which will be there in the phosphotransfer event. So, now you can imagine how things will move forward, that upon sensing the ligand, the histidine kinase is activated. This kinase now phosphorylates the histidine residue on the kinase or the receptor. Internally, phosphate is transferred to the aspartate residue. Now, this aspartate residue on the receptor will transfer phosphate onto the histidine residue of histidine phosphotransferase. This histidine phosphotransferase will be able to transfer the phosphate to the next aspartate residue which is present in the response regulator. As we saw in previous slide, the response regulator could be the simplest one, which has nothing but this aspartate domain which receives the phosphate group, or it can have an additional domain which can have an enzymatic activity, or it can have transcription factor activity, right? So, you realize that here it's not just one step transfer of the phosphate group from one histidine to the another aspartate residue. Rather, there is a sequence of event where histidine is involved, then next aspartate is involved, then next histidine is involved, and then finally you have the aspartate residue. So, here it's not exactly a phosphotransfer, rather it is called phosphorelay. So, your canonical two-component system will be identified by phosphotransfer and no requirement of histidine phosphotransferase like molecule, and the hybrid two-component system, or if you say three-component system, that will require the use of the histidine phosphotransferase like molecule, and there the phosphorelay kind of event happens. Now, in simpler terms, as we saw, if you have hybrid two-component system, you will have the sensor kinase, you will have histidine phosphorylation, then you will have phosphate group moving from histidine to the aspartate. This will require another histidine residue for phosphotransfer event, and this is where you will have HPT like molecules, and from here it will go to a aspartate residue in the final effector molecule, and these will be your response regulator. So, what you would notice that there could be multiple molecule in this pathway, and we will see some examples that where multiple different types of response regulators could be there. Eventually, the response will be mediated by a response regulator which will be called master response regulator. So, let us see what all happens in such scenario. So, let us take an example of signaling in where you have some sort of chemo attraction event going on. Here we will take example of how bacteria senses the presence of this molecule which is called cyclic di-GMP. And this name itself suggests that what will be the origin of this molecule. So the response regulator in this case will have this additional domain which is called GGDEF motif containing domain, and this motif containing domain, they have this catalytic activity which is referred as diguanylate cyclase activity, which means that they make linear nucleotide into a cyclic nucleotide, and that is why they will generate cyclic di-GMP. So what this domain containing protein as a response regulator does, it utilizes 2-GTP molecule through the activity of this GGDEF motif containing domain, it converts it into the cyclic di-GMP. If I can simply draw in cyclic di-GMP, and if I write GMP and GMP, so you will see that there are bonds which are formed between the two GMP molecule, which makes it cyclic di-GMP. What this molecule does, this is a big chemoattractant which will ensure that bacteria prefers a sessile lifestyle which will induce the biofilm formation. Okay, so this presence of this GMP or cyclic di-GMP will be sensed by the other bacteria in the vicinity, and they will come together and then they form the film. As soon as you have high levels of cyclic di-GMP, you will see the bacterial mobility is inhibited, right, because they now are getting into the sessile nature or in biofilm formation mode. This will also inhibit the virulence because the freely moving bacteria or microbe will be more approaching to the host cell and invading them. So presence of cyclic di-GMP makes the bacteria or the microbe less infectious. So you can understand that bacteria may not prefer this situation to be less infectious. That's where it will need a dynamic regulation on the levels of cyclic di-GMP, and this is where the another set of enzymes which are called phosphodiesterases come into picture. So you can think of one response regulator which catalyzes the formation of cyclic di-GMP, and the other response regulator which breaks down this cyclic di-GMP. And there are two different types of these response regulators. If I say response regulator 1 and response regulator 2, one of them will break down this cyclic di-GMP at one point and generate a linear GMP like molecule. For further breaking this down into the single GMP like molecule, you have another response regulator which contains this type of motif in their catalytic domain. So the first one, which has EAL motif in their catalytic domain, will break down cyclic di-GMP into the linear di-GMP. Then you will have the other HDGYP like of motif containing response regulator, which will now break down this GMP thing and generate 2-GMP molecule. So you can understand that given the environmental cues, the bacteria will synthesize cyclic di-GMP, and when the situation becomes more favorable, so that there is no stress, there is no resistance that need to be put forth, then it will break down the cyclic di-GMP and generate the molecule which will help in the process of more motility and more infection. Let's see that. Basically what I have told you here is that you will have response regulators of different types. If I can say, they have catalytic domain, one which allows cyclic di-GMP formation, and the other two will break down cyclic di-GMP. And the process is linear. 2-GTP is utilized to generate this molecule GMP, GMP, a cyclic nature, and you will have two response regulators. One will break down into GMP, and this will be breaking down into 2-GMP. So, let us see that in action. So, basically what happens, that GTP GMP dynamics is being strongly regulated in response to the environmental cue. When there is some sort of threat to the environment, whether it is nutritional threat or presence of another microbe or maybe parasite which may eat or feed on bacteria, this GTP is converted into cyclic di-GMP, which will be serving as an effector, which will be serving as a signal for these effector molecules. This will promote the biofilm formation or sessile life form, and if this effector is going to be operating on another response regulator which will be saying inhibit this free living form. So, together the dynamics between the biofilm form or the sessile form and the free living form is going to maintain the balance between free living and biofilm form or the quorum sensing form of the bacteria and give the resistance. So, what you notice here is that there is some feedback which is operating on this pathway. Cyclic di-GMP is produced, it will be broken down into GMP by EL phosphodiesterase. Then this linear di-GMP will be used by the HDGYP phosphodiesterase to generate two linear GMP molecules. What you notice here is that there are inhibition or feedback control on that. High levels of cyclic di-GMP will inhibit the enzyme which was helping in the formation. So, this is a negative feedback which is given to the bacteria that signal is enough. Also, the presence of free GTP will be giving inhibitory signal to the diesterase, so that you allow more and more cyclic di-GMP formation. So, together you will notice that the dynamics between GTP as a linear form converting into GMP first through an intermediate of cyclic di-GMP is tightly regulating the life forms of the bacteria. So, what we understood today, and it can be summarized, that the two component system in bacteria and microbes could be canonical type, where you have phosphotransfer event going on, or you can have hybrid type where phosphorelay kind of response is going on. The response regulators which are downstream in such scenario, which respond to chemoattraction, they help regulate the GTP GMP dynamics to mediate life forms which is sessile or the free living form, and this becomes very critical, and there are feedback regulations. On the enzymes, whether it was a cyclase enzyme or phosphodiesterase enzyme. So, you understood that how two component system is important for mediating chemoattraction, life forms in the bacteria. Thank you.